Okay, guys, good morning. Here's what we're going to do today. We're going to take a look at some of the external morphology of the brain. Here's a human brain right here, maybe a little bit smaller than normal, but as we talked about in class, probably 3 pounds of sort of a semi-gelatinous material. Different than this. This is fairly hard, but the actual human brain has more of a gelatinous material to it. So here's our model here. Here's the underside, the ventral surface, pituitary gland there. And this would fit very conveniently into the skull here. The pituitary gland, we'll talk about later, fits into this little indentation here, which is called the sella turcica or the Turk saddle. So amazing how we have this design, right? Not by chance, but a design that protects that pituitary gland as the brain is in the skull. Okay, couple brains that I have here, and both of which we're going to take a look at and dissect. Here's a sheep brain, and here is a horse brain, considerably larger. Sheep's not known for their mental acumen or cognition, right? Kind of a tiny brain. If you look at a brain that's inside a skull, if you were a neurosurgeon and you were to take off the skull cap or the calvarium, let's go back to this, you would not see a clean brain like this. We talked about this in class. There are three layers, three connective tissue layers that cover the brain, and we're going to identify them right here. This outer one, which is really thick and fairly stout. If I pull on this, it's a little bit tough. This is called the duramater, or the tough mother, literally. Okay. Beneath that, you have another layer called the arachnoid meninx, or the arachnoid layer. Arachnoid meaning spider-like. Spidery-like projections that actually, the underside of which is filled with cerebrospinal fluid. So in the actual brain, you have a cushioning of fluid here called cerebrospinal fluid that protects the brain. And then just beneath that, you have what's called the pia mater, or the loving mother, the tender mother. This is that little layer that would envelop the entire brain, almost a shrink wrapping, as we would call. Okay? So if I take some scissors, we're just going to trim off some of the duramater. Be careful as you do this, because if you rip it off too quickly, you're going to rip off some of the important structures, like perhaps the pituitary gland, or under here the optic nerves forming the optic chiasm. So you got to be careful when you're down here. But this is the duramater. I'm pulling on this probably as hard as I can. It feels like a thin sheet of plastic. So there is some good protection externally for the brain. Right here, you can see that. I can tear it, but very hard to pull apart. Pretty good tensile strength there. So I'll cut a little bit more of this. And you'll notice that there is a thickening here that I'll try to cut out. Hopefully it's not going to take that much time. But this is called the falx cerebri. And essentially the falx cerebri, if you see right here, it's a thickening of that meningeal layer here. You see how thick that is? And this goes down into the longitudinal fissure. We'll talk about that. But the deep clefts on the brain are what we call fissures. So the falx cerebri is just a meningeal thickening that I'm cutting off here, revealing the two hemispheres. So I come back here, I trim off more of that. And now there's another thickening here that you can see, which is a thickening of the meningeal layer between the transverse fissure. Okay, so this is the fissure that delineates the posterior portion of the cerebrum from the cerebellum. Okay, so again, a thickened piece of material that's really hard to get off. And if you're not careful, you probably end up pulling off the entire cerebellum, of course, which would make it very difficult for this pig to have any adequate balance. So we want to try to leave that intact. So here's the tentorium cerebelli. Again, a really thick piece of duramater. Tentorium means tent, cerebelli having to do with the cerebellum. So it's like a tent skin, literally, that is between that. So now I've revealed the two hemispheres of the cerebrum. Two hemispheres of the cerebrum here, two hemispheres of the cerebellum. As we look on the underside of the brain, or the ventral surface, I'm going to cut off a little bit more of this duramater right here. So we're getting down to where we could probably see the pituitary gland here, also the optic nerves here. If we were to trace this out, there's no eyeball here. But if we look right here, this grayish brown material right here, these are the extraocular muscles which would have attached to the sclera of the eyeball. So the pig would have used or the sheep would have used these muscles to actually control the movement of the eyeball. The eyeball is gone, so we're just going to snip that right there. And you see that the eyeball also has a lot of protective adipose for cushioning right here. And if you look right here, here is the optic nerve. This would have led to the retina, or the retina with the photoreceptors would have taken the information in. This coalesces then into the optic nerve, which travels through here, which travels here, some of which crosses over to the opposite hemisphere. Some of the information stays on each side. So we'll talk later about the eye, but you have input from both retinas providing depth perception and a good field of vision right there. Let me show you something interesting right here. As I pull this forward, I'm going to pull both of these off. You'll notice there are two little peanut-like structures here, very well developed in a pig. And these are called the olfactory bulbs. The olfactory bulb will take information from sensory receptors which are in the mucosa or in the nasal area. So if this sheep were to smell, all right, air would be carried into the nasal cavity. Little particles of material would be picked up by receptors in the nasal mucosa. These little fibers would coalesce, travel through the skull in what's called the cribriform plate. I'll show you that in a minute. These then would travel to the optic or the olfactory bulbs. Olfactory bulbs then travel back to the cerebrum where this information is integrated. I want to show you just the difference here in a human. Notice the size of the olfactory bulbs, completely tiny. We think we have a good sense of smell, but comparatively, our sense of smell or olfaction is really limited compared to other animals like the sheep. And in fact, if I show you the horse right here, look at the size of these guys. So an animal, let's say a male, say during breeding season, may use the incredible sense of smell they have to detect whether or not a female is in estrus or ready to breed. So incredible sense of smell here. Okay, let's go back here, and I want to just trim off this kind of mass here, which would lead in from the nasal mucosa. So there you go. You can really see the olfactory bulbs right there. I'm going to take off a little bit more dura mater here, and the dura fully wraps around the optic nerve. And if you remember in class, we talked about the fact that there are 12 optic nerves. You got the olfactory, you got the optic, a number then, many of which originate from the brainstem down here. It's going to be hard to see each of the 12 pairs here. It's really difficult. It's a lot easier to look at a model or to look at a book, but at least we can identify a few of them. Do a little trimming here, and probably as much as I want to take off, maybe a bit more, to see if we can take a look at the pituitary. And I'll point out the pituitary right here. We said this is the master gland of the body. Minute amounts of hormones are released from this, travel into the bloodstream through an endocrine function. Ductless glands secrete right into the blood. These hormones then are affecting target tissues in other parts of the body. But the pituitary is also affected by hormones released from the hypothalamus. So the endocrine system is one of the most complicated systems in the body. We're not going to talk about the entire system in depth, but when we talk about hormones and organs, we will occasionally talk about the endocrine system. Okay, let me see if I can cut this here. I think I just did a little bit of excessive trimming, and I kind of cut the chiasm, but we talked about the optic nerve. So the optic nerve from the right eye would come in here. And chi means cross. This is the optic chiasm or the crossover. So some of the fibers stay on this side, some cross over to the other side. So each visual cortex on each hemisphere, which is located roughly here, is going to receive input from each eye and then form a good image that the body can then interpret, or the brain can then interpret. And this is just more tentorium cerebelli that I'm pulling off here. Okay, so that's a decent job. I just kind of section this. Okay, we can snap that back together. That's easy. All right, Let's take a look at the superior surface. Okay, we're back. Let's take a look at the top of the brain, the dorsal surface, the superior surface. We can see that there are two hemispheres here, a left and a right hemisphere. I know in the human brain, it's roughly 80% of brain size. We have if we turn this over ventrally, we have the cerebellum, we have the brainstem, and we have another part called the interbrain, which we've already talked about. We have to cut the hemispheres in half really to look at that. Same basic design for the horse brain. But what I did want to show you here in the horse brain is we took off the dura mater right from the sheep brain. The dura is still here on the horse. I haven't peeled that away. But beneath that, you can see the little spider-webbed appearance here. This is what's called this is the subarachnoid space. This would be filled with cerebrospinal fluid, right, in a live horse. So this is going to be providing a lot of cushioning here. And you can just see how thick that dura is there. And then if we were to peel this away, you have that really thin layer called the pia mater. All right. Let's do a longitudinal section here. I'll take out my scalpel. We're going to cut through this, and I'm cutting down through the longitudinal fissure. It goes down, it reaches down to a certain point at which there is a connection between the left and right hemispheres. This connection is a white fiber commissure, as what it's called. And it is specifically called the corpus callosum, all right? So what I've just done is I've transected the brain. A couple things that I can point out right here. This is the corpus callosum right here. I just severed that. So in a sense, if this were separated in a human or a sheep or pig, for that matter, a horse, this would prevent the movement of information from one side of the hemisphere to the other. Can you live with a transected corpus callosum? It's called a callosotomy. Absolutely. But you might have a little bit of issue with certain types of perceptions, okay? For instance, in a human, the speech center is located roughly here in the frontal lobe. So hard to say. People have had this done. This was done in the past as a therapy for epilepsy. This was transected, preventing impulses from going from one side of the brain to the other sort of haphazardly. It likely cured the epilepsy, but it probably left some deficits in terms of function. If I open this up right here, you can see that space there. This is called the lateral ventricle. Here's the left lateral ventricle. Here is the right lateral ventricle in here. This is filled with cerebrospinal fluid. If you notice here, it's hard to see. We'd have to really magnify it, but I think you could just take my word for it. On the roof of the ventricles here and here, as well as on the roofs of the third and fourth ventricles, right, there are little groups of tissues called choroid plexuses. And these plexuses produce cerebrospinal fluid. Here's what it does. It's produced in here. It flows down through a series of different third, fourth ventricle, and then finally some of it circulates down here into what's called the central canal, down into the spinal cord, and then comes back up. So some of it goes down to the spine. Some of it is actually underneath the arachnoid layer, and this is going to provide some cushioning from the brain. It's a lot like blood plasma, a little different in some of the composition. Okay, let's take a look at the cerebrum. You'll notice that there are a lot of foldings on this, and as we talked about in class, this is to increase the surface area. Collectively, these foldings are called convolutions. If we look at a raised section, this is called a gyrus, or gyri plural. If we look at a nook or cranny, or a deep area, this is called a sulcus or sulci. So even the pig brain, right, or sheep brain, and the reason I'm interchanging pig and sheep is I get either one or the other, and they're basically the same in appearance. So with this sheep brain, even a lot of convolutions, right, for the condensing of neural material. Horse also has quite a few convolutions. Now, in terms of development, human brains are much more well developed. But I did want to show you this. If we take a scalpel and I trim off the outer millimeter, just the outer few millimeters, what I've just done is I've taken off the cerebral cortex. So the cerebral cortex would be comprised of cell or neuron cell bodies. called gray matter. The rest of this is white matter. The white matter that you see here would be axons that project down into the rest of the hemisphere. So if you were to trim off all of the cortex here, right, this animal would not have any function, because the functional part of this is located in the outer part of the brain called the cortex. So if an animal is decorticate, that means they have no cortex, they're not going to have this type of function. All right. Quickly review some lobes. I think you guys know these. Here's the frontal lobe. This would be roughly the area of the parietal lobe here. Temporal lobe is located laterally. The occipital lobe, posterior right here, which is where you have a visual input. And then there's another lobe which would be buried roughly here, kind of at the junction of the frontal, parietal, and temporal. That's called the insula. So basically, if I take this probe and put it into the brain right there, the tip of it is probably where the insula would be. Not a lot known about that, probably related to memory. All right. Guys, if we go back to the brain to look at the functional and anatomical divisions, I'm going to take this horse brain. I'm going to make a, again, I'm going to cut through the dura mater, through the corpus callosum. I'm going to transect the cerebellum as well, and the brainstem. So I can open this up. We'll have a little bit bigger image to look at. Okay, so here is your horse brain. All right, here's the horse's corpus callosum right here. Lateral ventricles are there, and here we just cut through this round, kind of round area right here. Is part of the thalamus. So this is the thalamus here. Now, if you're wondering where the other areas are, roughly thalamus here, hypothalamus would be located down in this area here. Hypothalamus, remember, has a number of different regulatory centers. The epithalamus is located here, epi meaning upon, so the epithalamus right there. And then if I turn this over, I can show you where the pituitary is on the horse. That's roughly right there. Okay, so we looked at the cerebrum. We just identified this area here, the thalamus, hypothalamus, epithalamus, and the pituitary, which are regarded as the interbrain, or remember, the diencephalon. Okay, and then I've also made a transection through the brainstem. Okay, brainstem is made up of three things. You have the midbrain, the pons, which is kind of a rounded area, and then you have what's called the medulla oblongata. The medulla oblongata then becomes continuous continues on as the spinal cord. This then exits the skull through the foramen magnum. Okay, not to be gross, but if I were to stick this brain inside there just like this, right, the medulla right here would exit through the foramen magnum and then become the spinal cord. So let's identify those parts again. Here we have the proximate midbrain here, the rounded area called the pons, and then the medulla oblongata right here. A lot of the cranial nerves, remember the 12 cranial nerves we talked about, would emanate from here. And what's interesting is that if an individual were to lose his entire cerebrum and the rest of the brain and only have the brainstem intact, that individual could likely survive in, I guess, what's described as a vegetative state. They would be able to respire. Their heart rate would be controlled by this part of the brainstem. That brings to mind Mike, the headless chicken that we'll talk about later in class, that was being prepared by the farmer. He cut the axe. He kept it pretty high, so he took off the cerebrum and other parts of the brain, but he left the brainstem. So this chicken lived for, I think, longer than a year without a head, literally without a head. And because of the functioning brainstem activity right here, okay? Here we have the cerebellum. And if there's one thing you remember about the cerebellum is that this part of the brain is involved in the integration of motor movements. If you lack a cerebellum, you would not have good balance. Your balance would be thrown off. Now, granted, balance has to do with the inner ear as well, but the integration of motor inputs takes place here. So we cut this in half. What's interesting is you see this little white tree-like structure in here. That's called the arbor vitae. It's called the arbor vitae right here. It looks almost like a mini cerebrum that's been transected in half. Okay, so hopefully, In looking at this video, you'll get a better idea maybe of what you'll be seeing when you do your dissection, and that'll help you better understand that. And if you have any questions, we'll talk about that in class.